Alright, this lecture is on Chapter 12 in the Chemistry and Context 6th edition. Chapter 12 was the student-selected chapter in our course, and the focus on this chapter is genetics and the chemistry of genetics. How close to designer babies are we? The first draft of the human genome was completed in 2000. What have we learned from this? What are stem cells and what is recombinant DNA? So what are we made of? I'm sure you've heard of DNA, especially in some of the recent trials. All genetic information is stored in the nucleus of the millions of cells in the human body. Each nucleus contains chromosomes, 46 compact structures, intertwined molecules of DNA, and about 30,000 genes, components that convey one or more hereditary traits. DNA is a special template written in a molecular code on a tightly coiled thread that carries all genetic information. Alright, so the components of DNA are phosphate. Now we've talked about phosphate in chemistry. It's a polyatomic ion. It is written PO4 3 negative. And we can see there's our 3 negative, 1, 2, 3. And the way we could do that was, if you go back and look at Lewis structures, this would be the Lewis structure that gives us the lowest formal charges. And remember, these formal charges have to equal the charge on the polyatomic ion. So chapter 12, we're starting to take all the information we've learned in this course and combine it together to bring about an application to biology. Alright, so we've also talked about in chapter 11 on nutrition, sugar, CnH2n, O-N, where N can be different numbers. And here's the question. If you remember from chapter 11, is this a monosaccharide or a disaccharide? You should say monosaccharide. Remember that disaccharide is two of these ring structures connected together. And then we have what we consider the base of the DNA, and there are 4 bases. Here they are: adenine, guanine, cytosine, and thymine. Now, what we're going to find out is that these bases occur in pairs, and we'll call them base pairs. And instead of pronouncing each one of these, sorry about this, instead of spelling out each one of these, we give these symbols: A, G, C, T. And if you've watched some movies in the past, maybe you've seen the movie Gattaca, where your profession is determined by your DNA. And that's why they call it Gattaca, because you can spell Gattaca out of these bases. Alright, so what does each one of these components of DNA do? Well, the phosphate group can be acidic or basic depending on how many hydrogen atoms are attached to it. As an acid, we would know that a hydrogen would be attached to each one of these oxygens, and that's where we get the chemical formula for phosphoric acid, H3PO4. Phosphoric acid. Base pairs can also either gain or lose a hydrogen. They show an example in the book that the base could gain a hydrogen ion depending on what the environment is. If it's acidic, and we get NH3 positive here. And you can see that happen. Could happen here as well. Could happen here as well. Could happen. Let's see. On this one, could happen here. No, not there. Let's see. Could happen here and open up the ring structure. Or not the ring structure, but open up the double bond on the oxygen. So a combination of all three of these components, a phosphate, a sugar, and a base, is called a nucleotide. All right, a nucleotide. When we have a phosphate, a sugar, and a base together, that makes up one nucleotide. Now, something to remember here is Chargaff's rules. And Chargaff says in every species, the percent of adenine almost always equals the percent of thymine. Similarly, the percent of guanine always is essentially identical to the percent of cytosine. So that's what we call these base pairs, that generally when guanine is present, then adenine is present in equal amounts. So that's one base pair. And then below the line here, the cytosine and thymine make up another base pair. Now the advantage of that is we don't have to be able to look at every base pair. Once we know that one's present, we can assume the other is present. Alright, so here's our nucleotide. We have the phosphate group, we have the sugar, and we have the base. Any of the four bases can be used to form a nucleotide. Covalent bond, we talked about that, the sharing of electrons between the phosphate group and the carbon or the sugar. We look at it as a whole. And then another covalent bond here between the carbon of the sugar and the nitrogen of the base. So what does a DNA segment look like? Well, here's a picture of it. It's also in your book. If you're following along in your book, this would be on page 501. All right. So we can see there's one unit. There's another unit. There's another unit. And there's another unit. Phosphate groups responsible for linking each nucleotide. Phosphate. Notice the phosphate is the link between this base 1 we'll call it, and base 2 here. Or base 2 and base 3, or base 3 and base 4. Phosphate group reacts with an OH group present in the sugar. So notice that's where the linkage occurs. It's between the OH group of the sugar and the phosphate group. So you should know Chargaff's rule. So what this does for us is we don't have to identify every individual base within the DNA. Once we know that one base is there, we know the other base is present. And here's some different percentage base compositions of various species. All right, so the double helix. We hear that a lot. If you're following in your book on page 503, they show us a picture of a double helix. And the double helix was discovered in the 1950s. And again, this is the thing about history. You probably think of Watson and Crick when we think of the double helix of DNA. They were awarded the Nobel Prize in 1962 for the discovery of the double helical structure. But let's not also forget a British crystallographer by Rosalind Franklin, who through X-ray analysis or X-ray scattering noticed the distance, which can determine the distance between atoms, was able to determine that there was this structure that we later Watson and Crick coined as DNA. So Rosalind Franklin, Watson and Crick. Using Rosalind Franklin's X-ray diffraction data, Watson and Crick proposed a molecular model for DNA. So there's our nice picture of DNA. So here's what we've got here as well. Guanine and cytosine. So here's our base pair here, and we said those occur roughly equally. And what holds these two base pairs together, or their interaction between the two base pairs, they show by these dashed red lines. That's hydrogen bonding. Now we've talked about hydrogen bonding before when we talked about Lewis structures in water. We have something like water, and actually let's do it this way. Let's make it look more. Remember that water takes on a tetrahedral framework. There's a lone pair here and there's a lone pair up here. And if there's another water molecule nearby, we could show that like this. And then maybe a lone pair coming out through here and a lone pair going back through here. There is an attraction between this oxygen and that hydrogen, which we show as a dashed line. That's called hydrogen bonding. Now it's a small force, but it is the hydrogen bonding in water that causes water to be, in the form of a solid, ice, to be less dense than water in the form of a liquid. Now that's really important. We've talked about this before, that ice floats, and that's important. If ice floats, it can melt off during the summer, and we get that recycling of the water. Whereas if ice sunk, the lakes would be froze over most of the time because there wouldn't be enough sunlight getting through the surface of the water to melt all the ice. So this hydrogen bonding is also responsible why water boils at such a higher temperature than, say, something like hydrogen sulfide. And hydrogen bonding occurs between hydrogen and the elements either oxygen, nitrogen, or fluorine that have high electronegativities. So this hydrogen bonding helps keep the two helical structures together, and we get the double helix. So again, we're bringing information that we have learned from previous chapters. All right. The process by which copies of DNA are made is called replication. So we can see here that the original DNA strand up here is becoming unwound and separated into single strands. And the only thing that will attach to the guanine is the cytosine, and the only thing that's going to attach to adenine is the thymine. So we're replicating here. When these break, the next thing this A is going to attach to is another T. Alright, so we're replicating the strands. These are called daughter molecules. You got the old strand that was left over on this side, and you've got an old strand left over on this side, and then here are your new strands here and here. So don't forget the term daughter molecule. So the two unwind, they form complementary portions separate, and then that gives us the template for the synthesis of a complementary strand, and we get replication. The result is two complete identical DNA molecules. Now, what if we introduce some chemical that messes this up, that starts to change the strands, or starts to change these interlocking mechanisms on each piece? What would happen? Well, we would get something that's not identical, right? And that non-identical strand may be beneficial or not beneficial. Okay? There are 3 billion base pairs in each human cell. 3 billion. 3 times 10 to the 6th. Base pairs. Remember, these bases occur in pairs, and there's 3 billion of them in a cell. Okay? So these base pairings convey the mechanism of how genetic information is expressed, and that expression is seen through the proteins, the bases. So through the DNA in those base pairs or bases, we see that gives us the characteristics of individuals and also inherited illnesses. So some inherited illnesses would be like genetic diseases, maybe like cystic fibrosis or polycystic kidney disease or type 1 diabetes. All of these things are related to, are thought of as inherited illnesses through DNA. All right, we've seen proteins before. We know the key component of proteins is the nitrogen group. We know that the key component of the fatty acids, or not the fatty acids, but the carboxylic acids that we saw in the previous chapter with the proteins, is this carboxyl group, and then we have a hydrogen and a side chain. All right, so here are amino acids. That's what I'm thinking of. Yeah, I don't know why I was thinking fatty acids. So amino acids. So again, back from chapter 11, we have proteins, and those proteins, when they become protonated with this acid group, the carboxyl group, they become amino acids. So there are four amino acids here: glycine, alanine, aspartic acid, and phenylalanine. So these amino acids are essential for human growth and development. And the only difference between these four amino acids is the R group, the side chain. And this one, it's a hydrogen. This one's a methyl group. This one, it is a acetyl group. And then the last one, it's a phenyl group. There are 20 naturally occurring amino acids that make up proteins. Now some of those amino acids, again from chapter 11 about nutrition, some of those amino acids are essential. They're required for us to grow healthy bodies, and some of them have supporting roles. So two amino acids can link together. Here's the carboxyl group of one amino acid, and here's the carboxyl group of the other amino acid. So we lose a hydrogen here, or actually an OH group here, and we lose a hydrogen here, and what do we get? Hydrogen plus OH, the hydroxide, gives us water. And these two amino acids bond together, and we call that a peptide bond. Again, this process can repeat itself over and over, creating a peptide chain. And once the amino acids are incorporated into a peptide chain, they are called amino acid residues. Codons, how are they relevant in genetic expression? Okay. A codon is a sequence of 3 adjacent nucleotides, and they determine the insertion of specific amino acid during protein synthesis, or they signal the start and stop of a protein synthesis. So the codons is what helps us see the sequencing, or determine the sequencing. So in this case right here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9 base pairs. Remember, base pair is 2 bases together. All right. And this sequencing, this is codon 1, GTA, codon 2, AAA, and codon 3, GGC. All right. So this sequencing determines where to start and stop in the process of making a new or showing how these chemicals would, or how these pairs would make an inherited trait. All right. So the order of the bases in DNA determines the order of the amino acids in a protein. There are 20 amino acids present in the proteins. The DNA code must contain 20 code words. Each word represents a different amino acid. Okay, groupings of 3 DNA bases, codons. All right, the primary structure of a protein is its linear sequence of amino acids. Again, alanine, glycine, so forth. And the location of any of the disulfide bridges. All right, the sequence characterized by the amino terminal N-terminal ammonium or the NH3 positive ion at one end and the carboxyl terminal of the other end. So we saw that. We saw where this and this interact with one another, and we get the water and we get the peptide bond. All right. So a lot of times when we're looking at enzymes, we want to know what the structure looks like, the overall structure. And here we look for these terminal ends, carboxyl group and the N-terminal end. And then knowing where the terminal ends are, everything else in here, we're more interested in what the three-dimensional structure looks like. So we get this ribbon type diagram. All right. So most proteins contain one or more stretches of amino acids that give rise to a characteristic three-dimensional structure. The most common of these is the alpha helix and the beta conformation. Telephone cord represents the nature of the secondary structure. So you got the structure at the atomic level. That'd be the wires and the rubber molecules that make that telephone cord. But overall, if you look at a telephone cord, it's this helical structure. That's our secondary structure. The tertiary structures is when that's all, that telephone wire, it's not laying in a flat line like that it gets all tangled within itself. And it becomes, when we look at it three-dimensionally, that's called a tertiary structure. Like this, tangled up phone cord. So you could think of it this way. If these are the ends of the sequence, there's the carbon terminal and the nitrogen terminal. All right. So there's our entire amino acid sequence. All right. But in real life, it's not going to be laying in a straight line like this. And in fact, the tertiary structure determines a lot of its chemical properties and its physical properties. So in reality, the structure itself is three-dimensional, and it is a tangled mess. But we still can determine where the terminal ends are. That's helpful. And then we also see this three-dimensional structure that's inside the boxes, which gives it, again, its properties. In fact, if we look at something like a denatured protein, that's where you've got a sequence of amino acids that make up this protein, and it's got certain chemical and physical properties. And then when we denature it by heating it or maybe adding alcohol to it or something like that, it changes its tertiary structure and it's no longer got the same chemical and physical properties. Classic example of that is egg whites. When you take an egg out of the refrigerator, the egg whites are runny, they're clear, but when you heat them up, those proteins start to become denatured, and we get that white solid egg white. All right, so the function, this is what we just said, the function of the protein is dependent upon the shape of the three-dimensional structure. Small changes in the sequencing or the primary structure will cause dramatic effects in the three-dimensional structure and its properties. So the example that they give in the book is sickle cell anemia. Normally our red blood cells are round, but due to amino sequence errors we get these elongated blood cells, and we call this sickle cell. And because they have these abnormal shapes, they can't pass through the tiny openings of the spleen. They start to clot together. And these cells are destroyed and we get anemia, which means we don't have enough red blood cells in our body. And because we don't have enough red blood cells, people who have sickle cell anemia can have a lower blood supply for their body. Now, maybe you've heard about this, that their thought is maybe this genetic coding that makes sickle cell has an important effect for people who are from around the equator where malaria is prevalent, and sickle cell helps reduce the spread of malaria within the body. So it may be that it had a genetic benefit at one point, but overall we see it as a problem in modern day society. All right. The Human Genome Project, maybe you've heard of, again, Crick and Watson. Maybe you've heard of somebody who's worked on the Human Genome Project. The one that comes to mind is Craig Venter. He was the one to sequence this, the human genome with private money. And on June 26, 2000, it says scientists announced the rough draft of the project to decode the genetic makeup of humans had been completed. And again, what does that do for us? It allows us to start searching for those genetic, start looking at those genetic codes and see how we can manipulate them for our benefit. Maybe you have a genetic disease. Maybe we can fix that genetic disease through some type of recombinant DNA. Or one of the things that has been talked about, especially like here in Lexington, Kentucky with Alltech, is we can change the DNA of a yeast cell to be able to break down cellulose and generate ethanol, and then we can use the ethanol to power our vehicles. So we have to program the DNA in a yeast cell to specifically break down maybe the cellulose of wood products, or maybe the stalk of corn or something like that. All right, so what is genetic engineering? A representation of genetic engineering. So we have DNA from a gene source. We restrict the enzyme activity, so the gene fragments. All right. We insert the gene into a vector. So you have this gene here, and what we get is, here is a cell. Well, here's a bacterium. Yeah, here's a bacterium. Here's the chromosome. Here's the gene vector that we've modified, and there's the protein. So in this case, we're cloning the bacteria. All of these genes that were in the gene vector come out over on one side, and the proteins come out over on the other side of the bacteria. Now, there has been talk about using this type of technology also to make alcohol for biofuels. So, you have two things that come out of this modified bacteria. On one side, you get the genes harvested: plant resistance, gene therapies, DNA fingerprintings, diagnosis of genetic diseases, animal growth. That's with the gene. On the other side, you get these proteins that could be used for vaccines, insulin, human growth hormone, interferon, and tissue activator. So, a recent article I read was where they would take E. coli, which grows fairly quickly, and modify it so that it produces alcohol. And we got to produce a lot of alcohol if we're going to fuel all the vehicles in the United States. And because E. coli reproduces so rapidly, this process happens so much faster than other processes, and it doesn't require the amount of energy. The bottom line is, if we ferment alcohol, if we ferment corn to make ethanol, if we have to cook that to boil the ethanol off, well, that costs a lot of energy. We have to cook it. We have to add energy to boil the ethanol off. With this process, we could make the ethanol without that boiling component. So a lot of times with gene therapy or recombinant DNA, we are afraid that we will make make a mess of it. And so here's a chimera. It's a combination of a lion, a goat, and a serpent. And mixing up the DNA or messing up with the DNA, we might get something like this. If it would be viable, who knows? But I don't know if you've seen some other information about this, is that there's been a lot of talk that in the DNA of a chicken is all the DNA required to recreate a dinosaur. So there are scientists thinking about selectively turning on chromosomes within the to regenerate a dinosaur. All right. So, where do we use genetically engineered food? Agriculture. Actually, it's being used. We have genetically engineered corn, hybrid corn that allows higher production per given amount of acreage. Uh, there have been genetically modified potatoes. Um, so we are genetically modifying um, vegetables or plants. In a way, some people would argue that we do genetically modify uh, animals and things like that. Maybe not in the laboratory, but we definitely cross a poodle with a Labrador Retriever, and we get a new breed called a Labradoodle. No, we're not physically taking the DNA and manipulating it, but we are manipulating it by crossing those two animals, just like we cross a horse and a donkey and get a mule. And probably the biggest thing we hear about is cloning, that we could take the DNA, insert it in that vector, and come up with an exact copy of the original host DNA. So here's what Dolly was. We took the DNA of a sheep, put it in the the egg of another sheep, and we get a clone. You can also do it with dogs, and dogs have been cloned. The advantage of that is you get the same looking dog. Now, are they exactly the same? Same dog? No, they're not. I mean, that's where we get to start studying how the environment affects our growth. All right. So here they show the cloning of a sheep. So we got sheep 1. Remove the nucleus DNA from each cell. Remove the nucleus from an unfertilized egg. Plant that nucleus into this egg, and then electrical pulse diffuse the mammary cell in the egg. Culture the embryo. It starts to divide, and then implant that into another sheep, and we get Dolly, a clone sheep. So where do we go from here? What's your thoughts on genetics? What are the advantages and disadvantages? Is this process better or worse than the nuclear processes that we learned to do in the 40s and the 50s? What are uh, what are the um, what are the ethical considerations that should be thought of when we're doing these types of experiments? These are all things that you'll need to think about in the future and today. So I hope you enjoyed this student selected chapter, and we'll see you later. Bye.